Yes.
I'm sorry.
No, you're not. This is from your friends. So it's her birthday. You can hug her. Sing it. Happy birthday to you. Happy birthday to you. Happy birthday, dear Shree. Happy birthday to you. So capillary exchange. We've got passive diffusion going through water-filled pores that are actually in the plasma membrane of they're called channels though when they're in the plasma membrane, and then certain types of capillaries have little openings between the endothelial cells so that you have what's called a paracellular route for water-soluble substances to go through. And we'll see that on a later slide. The extracellular fluid is split into two sections. The fluid that's in the plasma and the fluid that's in your interstitial space. And those two equilibrate, and then the interstitial fluid equilibrates with your intracellular fluid. When you have fluids in the plasma that suddenly, or in the vasculature that suddenly increase or decrease, then you're going to get either a pushing or a sucking, driving force between the interstitium and the plasma. So we have this fluid pressure that is moving substances that are small enough through these open channels and then out or into the shared vascular or interstitial fluid. Lipid-soluble substances, they just go through the plasma membrane, don't have any, they go down their diffusion gradient, but they don't have any real barriers to diffusion. The plasma proteins generally are not escaping from the vascular system and out into the interstitial fluid. However, you have to, the reason I put that in red is not because I added the word, but I wanted to emphasize that this doesn't exclude plasma proteins from leaking out. If they're small enough, they will leak out. But normally the bigger proteins will not appear in interstitial space. So if there's a pathology, then you'll see more plasma proteins out in the interstitial fluid. So here, I've added some pictures here. So we have endothelial cells of your capillaries, and we've got these little pores that are in between the endothelial cells that allow for bulk flow of fluids to pull solute that's small enough along with it out into the interstitial space if the pressure, the water pressure in the plasma is higher than it is in the interstitial space. If it's lower, then there'll be a sucking pressure where fluids will return to the vascular system. Normally, this is occurring at the beginning of a capillary. You have more water. As the water leaks out, as it's flowing through the capillary, then the water pressure inside becomes less, and then there's a sucking pressure back in. But the pressure outside capillaries normally never exceeds the water pressure inside. But if you remember way back to last semester, when water leaves, then the solute concentration increases. And that gives you a driving force for water to come back in. So remember about the changes in concentrations due to something leaving a compartment. These big holes that I superimposed, those would be fenestrations or windows in the endothelial cells. You see that where you have to get proteins across, like hormones, or you see it where you want to filter things across, as in the kidney. And then we have small little channels that are membrane channels, and those also allow for ions to move. We learned about sodium and potassium ions that move through their channels, and water will be able to go through it. Sodium and potassium can go through the channel, water can go through it as well. And then we'll also have what are called large pores in between endothelial cells. They're really called discontinuous capillaries, where these endothelial cells don't even touch each other really. They have big gaps between them, and there's such big spaces that cells can just, white blood cells can easily get in and behind the endothelial cells, and then do a phagocytic job by looking at what's running through the capillary. They'll hang out outside here, but then through the openings, they'll be sampling the fluids and the plasma for pathogens. So large discontinuous capillaries will be in places like the liver, where you want to have this accessibility of the substances inside the capillary to phagocytes outside the capillaries. Here we are shown an example. We've got plasma proteins in the blood. Normally they don't leak out, but some small ones will leak out. And some are actually transcytosed out in certain areas. So proteins can get out. Certain hormones especially will be transcytosed out so that they can have access to their receptors. Then we have the small ions that will be able to flow either paracellularly or go through, not shown here, the channels that sodium will leak into a cell, potassium will leak out of a cell. And by the movement of charged particles, you can create an electrical gradient that will then either help or impede the further movement of ions. Anything that's lipid soluble or any gas, like CO2 and O2, they just diffuse right through the plasma membrane. Don't need to go, but they could go through the paracellular route. They don't have to go through the cell, but they have a huge surface area for their diffusion because they're not hindered by the plasma membrane. So they can go through the plasma membrane of the endothelial cells or through the channels. Any place where water can leak across, then ions can move down their concentration gradients. So it could be into the plasma, it could be out of the plasma. So here they try to show you the concentration gradient. So glucose will be higher in the blood, so they give you a broad side to this arrow, and then they'll be consumed by the tissue cells, so there'll be less of it in the cell. So this will give you a gradient for glucose to start to diffuse out. It can't enter the cell by itself, but there are diffusion proteins that will use either sodium or some other molecule to concentration gradient to then pick up glucose as something like sodium binds, then glucose will bind and it will get into the cell. O2 is diffusing through the endothelial cells, it's being consumed by the mitochondria in the tissue cell, and so there's a gradient for it to go in. O2's consumption and CO2's production, you have a reverse concentration gradient for CO2. High intracellularly and then lower in interstitial fluid and lowest in the plasma. It's also binding to the hemoglobin in the red blood cell, so it's not building up a concentration in the plasma. So it's leaving the plasma, going into the red blood cell, getting attached to hemoglobin, or getting converted to bicarb and disappearing magically because now it's a bicarb molecule. So that keeps the gradient for CO2 to continue to diffuse out of the tissues. So bulk flow is, the book says protein free. I put ish there because it does contain some proteins, small proteins. And those, as the fluids generally are leaking out into the interstitial fluid, then the concentration of solutes that can't get out is going to increase, and so eventually there will be a reverse concentration gradient for fluids to move. The fluid that doesn't come back into the capillary then will get picked up by the lymphatic system and brought back to the heart. So our blood volume isn't dropping because we can't get all the fluid back in before the blood gets back into the venules. So there is going to be some fluid that's not able to get back in the bloodstream in the capillary bed. But as we'll see later in the chapter, there's adequate lymphatic vessels there that will pick up that fluid and then return it to the heart, and then it will re-enter the vascular system. When we talk about ultrafiltration, we've got a hydrostatic pressure in the blood vessel that is pushing things out, and as that fluid is moving out, as I said, small solutes can go. And then this is taking fluids and small solutes out of the blood vessels, and then later on, as the fluid pressure goes down, ultrafiltration will decrease enough so that the solute concentration in the blood vessel then can suck blood back in, and that will be referred to as reabsorption. So we've got two tendencies that are in opposition to each other. A ultrafiltration, which puts fluid out into the interstitial space, and then reabsorption, which will bring fluid back into the capillary. So we call the forces that help with reabsorption the capillary oncotic pressure, which is that sucking pressure, because the solute is increasing in the capillary. And then there is a slight hydrostatic force that is out there in the interstitium, also pushing fluid back in. But that's very small. The majority of reabsorption is occurring because of this increased capillary oncotic pressure. And we'll talk a little bit more about what happens to the solutes that go out there. They're either consumed, so they go into cells because they're nutrients that the cells need and will take them up. Or if they're in excess of what the cell needs, or there's something that leaked out and is not used by the cell, then those usually are going with the excess fluid back in, well, not back into, but into the lymphatic vessel and then they'll be returned to the systemic circulation. So this kind of a frightening looking thing now that I've gotten a hold of it and added some more to it, but it's not that big a deal. Yeah. It's really an equation, and what I've done is pulled some things out and then put it together in a workable equation that will help you during exam periods. And I've also pointed out what their little characters mean, and I'm suggesting that although the book is using them, really translate them into what other books use. For example, this P here, it's not talking about hydrostatic pressure, but that P is talking about hydrostatic pressure. So how do you know? Is it a capital P or a small p? Did they forget to capitalize the P and it's hydrostatic pressure? Or is it the oncotic pressure of the capillary? So the pi is telling you it's oncotic pressure, but under stress, you think, well, am I supposed to multiply oncotic pressure by hydrostatic pressure? No. So I would suggest you just use capillary, CAP, to clarify for yourself that this is capillary oncotic pressure. And on here, they did use interstitial fluid, or IF, or interstitial fluid. And I oftentimes use ISF just because I'm used to doing that. But here we've got capillary, and you can put CAP there, capillary hydrostatic pressure. And we've got oncotic pressure in the capillary. And then outside, we've got a hydrostatic pressure due to the fluid in the interstitial space. And we've got an oncotic pressure in the interstitial space. So pi sub IF. And the text is saying it's 0, but I'm saying it's not, because some small proteins diffuse out. So it's close to 0, but it's not 0. And then if you are going into pathology, so you're going to be a nurse, you're going to be a doctor, you're going to be dealing with pharmacy, you have to realize that when pathologies get to the kidneys or get to blood vessels, then this number is never 0. So you have to remember, you can't just say it's 0 and ignore it, because it will become important when you have pathologies, and it will be important when you have edemas, tissue edemas. So make sure that you realize it is not 0. So over here on the arterial side, we've got some forces that are favoring ultrafiltration. We've got hydrostatic pressure that is 37, and we've got that pressing out. It says we have a 0, but it's really not a 0, so maybe it's 1, 2, or 3. So this is probably something more like 34 or 35. And then we've got an inward pressure that's due to the interstitial hydrostatic pressure, which is 1, and it is low. It is usually around 1 or so. And then we've got a capillary oncotic pressure, which is 25. And so as they're implying here, you have certain things that are working together to either move solute and fluid out or bring solute and fluid back in. So what I did up here is put the things together in an equation. So what's working together? You've got the hydrostatic pressure of your capillary, and the oncotic pressure of your interstitial fluid. Those work together to move fluids and solute out. So that favors ultrafiltration. It's working against the inward pressure, which is your interstitial fluid hydrostatic pressure and your capillary oncotic pressure. So this equation, if you remember it and put it down in this order, and you don't subtract in here and add out there, or put hydrostatic forces together and oncotic forces together in this equation, you will be able to determine, is the kidney working properly at the is the blood vessel working? It's going to work for kidneys too, so I'm just getting excited and jumping a lot of chapters ahead. So the same thing will come back, you use it again for kidneys. So if at the arterial side, this sum is larger than that sum, then you'll get a positive answer, positive X. It is, if that is excessive, you get edema. If you get a negative answer, then that means you're favoring reabsorption. Normally you don't favor reabsorption at the arterial end of a capillary. Normally that's going to occur over here at the venous end. So actually you would use this equation twice. You calculate what the net filtration pressure is at the arterial end, and it should be positive. It should be a small number so that fluids leak out and nutrients go with that fluid, but not exceeding what the lymphatics can then carry away. So edema occurs if you exceed lymphatic clearance, and if it's occurring the whole way along the capillary. So when pathologies come in, you don't get a negative number at the venous end. If you have nephritis, or you have an autoimmune attack in the kidney, those blood vessels become more and more leaky. And if you have a plasma protein problem because you're an alcoholic and you have liver cirrhosis, or you have liver cancer, or you have hepatitis, then you don't have adequate plasma proteins to reabsorb at this end, so you still are getting net filtration at that end. So if you get a positive number here and you get a positive number there, then you're going to be getting edema. You're not able to get that fluid back into the vascular system. So that could be a blood vessel problem. Either your vessels are more leaky than they should be, or they don't have the proper oncotic pressure, and so they can't keep fluid in the vascular system. And then we're going to see shortly that you can also have lymphatic problems, and the lymphatic drainage is blocked, and the fluid will then build up and you'll get edema, but it's not a blood vessel problem, it's a lymphatic problem. Okay, so the other thing that I saw that's wrong with what's going on inside the blood vessel is here. You have oncotic pressure is 25, and you have oncotic pressure there as 25. But if water leaves, no way is your oncotic pressure constant. It's got to go up. So you have to remember that sometimes there are problems with your figures. Make a note that this should be greater than 25. And in the examples, the numbers that I give you in the test that you have to pick out from your table and then put into the equation, this number, I will give you sets of numbers. I'll say this is your hydrostatic pressure at your arterial end, this is your hydrostatic pressure at your venous end, this is your oncotic pressure at your arterial end, this is your oncotic pressure at your venous end. And the oncotic pressure is not going to be the same. It should not be the same. If fluids leave, then the concentrations of solids go up. And that includes everything, including the plasma proteins. When I first looked at this, I was like, huh? What are they talking about? So I put some clarification for you. The text, if you read it, it helps you clarify it. This green line here is your inward pressure line. And this is your outward pressure line. They're saying that at the arterial end, where you have higher O2 concentration, you're going to have an outward net filtration pressure. So ultrafiltration is occurring at the arterial end, and it becomes less and less and less as you go towards the middle of the capillary. And then at this transition point, then your outward pressure is less than your inward pressure, and so fluids start to return to the capillary. So they're implying that more and more fluid comes back as you go closer to the venous circulation. I have a problem with that as well because your oncotic pressure is highest in the capillary at that point, so the most fluid is going to be coming back early and then less later on. So take this, the first part works fine, second part doesn't work very well. So don't concentrate too much on this side. Concentrate on that side, but remember that this equation on this side, on the arterial side, you should be getting a positive number, meaning you're favoring ultrafiltration. On this side, you should be getting a negative number, which means it's favoring reabsorption. I don't think you get more reabsorbed later. That, as you're diluting your oncotic forces, you're not increasing any hydrostatic forces. There's no way you can get larger amount later in the capillary. Okay, so what happens to the fluid that's not reabsorbed? That is going to be picked up by the lymphatic vessels and then return to the heart, and it goes back into the systemic circulation. So really the only fluid that is present out there in the tissues is enough fluid so that you have adequate water concentration, so that water's not being sucked out of a cell and water's not going into the cell. You want to keep that water concentration stabilized, and you want to keep the solute concentration stabilized outside in the interstitial space. So any excess water, any excess solute, you want it to be picked up by the lymphatic and then put back into the bloodstream. Also, you don't want the fluids to collect out there for a couple reasons. The fluid collects in your interstitial space, that means your blood pressure is dropping. And then you've got to do something about that. Either your heart has to work harder, or your kidneys have to work harder, or you've got to drink or you gotta consume whatever is missing and is in your interstitial space and not getting back into your systemic circulation. So your body has to work harder at some point if fluids are collecting out in the interstitial space. If fluids are collecting out there, then they're going, even if they're isosmotic to the cell, if the fluid pressure builds up, what's going to happen to the cell? It might not burst because it's isosmotic. The water's not entering the cell, but water is doing what to the cell? Huh? Yeah, compressing the cell. So it's squishing the cell, and the cell's not going to be able to function when it's all squished up and can't get things moving through the cell as it's supposed to be transporting through the cell. So lymphatics, that's what this vessel is. We see that it's not connected to anything. These are referred to as blind-ended capillaries. So they start in the periphery. They're ending essentially, but actually it might be better to say they're blind starting, because they start in the tissues and then they are their directional flow is back to the heart. They're referred to as blind-ended. They're not connected to anything. This is connected to the larger arteries. This is connected to larger veins. So it's a circuit. These are unidirectional. They start in the tissues and then they converge to form larger and larger vessels, but they all converge to then dump into the vena cava or brachiocephalic veins right before the blood goes back to the heart. So fluids that leak out on the arterial side will, if they're not reabsorbed on the venous side, then they will go into this lymphatic channel and then go back to the heart. So looking at the lymphatic capillaries, they're made up of endothelial cells. The endothelial cells kind of overlap each other, and they have, not shown here, but if you imagine that this arrow had an attachment inside, and then it was attached at its tail to connective tissue outside. As the fluid pressure builds up outside, it's going to, the connective tissue is going to start to get pushed apart by the fluid pressure, and that's going to tighten this arrow and then tug on it from the tail to the head. What that does is it pops these little channels open, and fluids then can enter the lymphatics. And when the pressure isn't high outside, the lymphatic vessels close. So they're regulated, they're picking up fluid when there's excess fluid to be picked up. If there's no excess fluid to be picked up, then their little openings are closed. So fluid pressure building up outside, you're going to have these tethering, anchoring filaments pop the endothelial cells open, and then the fluids are going to go into the lymphatic vessels. So that's what I put up here. You got an anchoring filament attached to CT, which is connective tissue outside. As the fluid pressure builds up outside, then there will be tethering that will pull the channels open, and then that will permit the fluid to come in to the lymphatic vessel. Once it's in the lymph, then the fluid pressure will just start the fluid moving backwards towards the heart. There are no smooth muscles to contract in the lymphatic vessels, so they're also relying on a skeletal muscle pump for the fluid to move through. What's not shown here is the lymphatic vessels have valves. So once the fluid gets beyond that valve, it cannot go backwards towards the blind end anymore. It's a one-directional valve system like in the medium-sized veins in the vascular system.
The lymphatic system consists of lymph vessels and lymphoid organs. Lymph capillaries thread through capillary beds. Fluid and plasma proteins that leak out of the capillaries enter the tips of lymph capillaries. The lymph capillaries merge into larger lymph vessels. Lymph nodes occur at intervals along the vessels. As fluid trickles through a lymph node, areas of white blood cells monitor it for invaders. If they encounter a foreign antigen, they call up a rapid immune response. The filtered lymph fluid passes into larger lymphatic ducts, which carry it back to the circulatory system. The tonsils, thymus, and spleen are lymphoid organs. Lymphocytes in the tonsils protect against inhaled microorganisms. Immature T cells differentiate and acquire their unique receptors in the thymus. The spleen filters pathogens and used-up blood cells from the blood and is a major site of antibody production. Now let's check what you have learned. Drag the text to the blanks to label these components of the lymphatic system.
Okay, you can do that on your own. So coming back to this picture, these lymph nodes, you have them at your elbows, you have them back of your knees, you have them down at your ankles. So not just up here in the inguinal region in the center. You have them around your breast, have them in your axillary region, a lot of them up your neck and around to the back. The spleen will have two different sections. One is called white pulp, which has white blood cells in that area that are looking for pathogens, and then red pulp, and that's looking for worn out red blood cells. So extra fluid, any leaky proteins, those will go through the lymphatic vessels. They have multiple areas where they can be checked for pathogen. So down in the legs, up in the arms, and then again at the axillary region, then again right before going into the brachiocephalic veins to go back to the heart. They got multiple sites where they'll go through lymph nodes in the head region and also in the thoracic region around the breast region as well. So you're repetitively looking for pathogen. And so if it's missed in an early lymph node, hopefully it's picked up by the later lymph node before it gets into the systemic circulation and then gets distributed back out to the tissues. So common places where you inoculate yourself. You put things in your mouth. You're always sticking things in your mouth that don't belong there. Your pencils, your pens, other things. You have your respiratory system where somebody might sneeze and then you inhale the pathogen. So you've got your adenoids up here that will look at anything that was inhaled through your nose, and then you have tonsils in the back of your throat that for anything that came through your mouth. And they're a little bit different than lymph nodes. They're called lymph nodules. We'll talk more about that in the lymphatic vessels. A lot of pathogens come through the GI tract. So in the mesentery that is holding your small and large intestine in place, you're gonna have a lot of lymph nodes. You have a lot of bacteria in your gut. You need that bacteria or you're gonna die. That's one of the reasons why people who don't develop bacteria, actually babies, if they don't develop that natural bacteria in their gut, they don't live very long. They're usually dead in a year or so because they need that. The bacteria help you to make things that you can't make for yourself. And they also keep your gut healthy. But they're not supposed to get out of your gut. But your gut sometimes gets leaky, and they do leak out of your gut. So you need the immune cells there to protect you there. Walking barefoot, stepping on things, doing things in the garden, other things with our hands. Sometimes we get things into our fingers, and that's one way we can inoculate ourselves with bacteria, and we can get funguses and things that route as well. Most bacteria are destroyed when they're going through the lymphatic system. So only the ones that can evade the white blood cells, and some of them can't. They have mechanisms to do that. And viruses can also evade. They have mechanisms to evade the white blood cells in your lymph nodes. If they do, then they get into the venous system, into the heart, and then they can get spread out into other organ systems. The lymphatic vessels also transport digested fats. Those fats are too big when they come out of the cells of the intestine they're not individual fatty acids or glycerols or cholesterol. They're in big chunks and, well, not as big as they were when you were digesting them, but big enough so they can't get in the blood vessels. So they use those big channels in the lymphatic system. They pop open, the endothelial cells get into the lymphatic fluid, and then the lymph brings it back to the heart, and then the heart can then distribute the fats throughout your body. If you don't know what elephantitis is, it's just a descriptive term for many types of problems. The legs get very enlarged and look like elephant legs, so they call it elephantitis. You can get it due to parasitic infections, people who walk around barefoot in the tropics. They may get parasites in their feet, and then the parasites start to replicate in the lymph nodes, and then the lymph nodes get blocked and fluid can't get past that lymph node. So a lot of times you'll see maybe the whole leg will be huge, maybe just down the feet or the lower leg, and then above can be normal. It depends on where the parasites are proliferating. So here it looks like maybe they're in the popliteal lymph nodes, so the leg on this side is swollen. It's actually edema, and it's called a pitting edema because if you press on it, it doesn't come back. Most edemas, non-pitting edema, if you press on it, it will bounce back. But here you have pressure on it and it stays indented in. So here it looks like the thigh region is enlarged too. I remember sitting in a doctor's office and I was feeling really lousy, but I happened to look over and I saw this lady with her leg up, and her leg was as big around as she was in the middle, and the other leg was normal size. So somehow she had gotten this parasitic or whatever type of infection blocking her lymph node in one leg, but the whole leg was huge. So it varies in how it's appearing. You can also, if you have had a mastectomy, a lot of times they'll remove lymph nodes in the axillary region because they're afraid that the cancer has metastasized. And they'll take out lymph nodes, and then do. They'll send them to the path lab. If they see any cancer, they'll come back and they'll take out more lymph nodes, and they'll do that until they run into normal lymph nodes. So when they do that in the axillary region, it's interfering with flow out of the arm. And so sometimes we get people with, well, females with, could be a male too, males get breast cancer too, but usually you see it in females. And the whole arm is just huge due to this tissue edema. If they catch it early enough, they can put compression on the sleeve on the person, and that helps to limit that. So lymph vessels, they're blind-ended. They will start in the head region, the hand region, the foot region, and then they'll join together and run through these various lymph nodes. So that will have a lot of white blood cells and also T cells and B cells in it to look for various types of pathogen. You'll have antigen-presenting cells there that can handle the active immunity. And so hopefully before it gets into the brachiocephalic vein and to the right part, you've cleared the lymphatic fluid of the invading pathogens. So anything like leaky gut or break in your skin or epithelial cells in your mouth that are broken down, where you are eliminating the barrier, then you have to rely on white blood cells at some point to clear that pathogen.
The lymph vascular system starts at capillary beds. Here, fluid that is leaked from blood capillaries becomes part of the interstitial fluid. Lymph capillaries have no obvious entrance. Water and solutes enter their tip at flap-like valves. These are areas where endothelial cells overlap. The lymph capillaries merge to become lymph vessels. These vessels converge into collecting ducts that drain into veins in the lower neck. Lymph nodes are strategically placed at intervals along the lymph vessels. As fluid moves through the nodes, infection-fighting lymphocytes monitor it for possible invaders.
It's a little repetitious, but that's okay. Okay, take a break.